This is Dexter. Ever since he was a little boy, he has always wanted to become a nephrologist. We see Dexter has fallen asleep studying physiology. And suddenly, it was clear how his urine was produced. Here, we see the kidney. They are essential in the urinary system and also serve homeostatic functions such as the regulation of electrolytes, maintenance of acid-base balance, and regulation of blood pressure, but most importantly, in producing urine. The functional unit which is important in urine production is the nephron. And with that, let's start producing urine. Glomerulus is a tuft of capillaries which is unique for its two arterioles. The blood enters the glomerulus through the afferent arterioles. Those that are not filtered in the blood will leave the glomerulus through the efferent arteriole. As blood enters the glomerulus, it is filtered into the Bowman's capsule. This Bowman's capsule is lined by podocytes that forms the visceral layer. It also has mesangial cells, which is important for structural support, secretion of extracellular matrix, and phagocytotic activity. Substances in the blood are filtered in the glomerulus according to its size and charge. Small molecules like water, sodium, chloride, potassium, urea, creatinine, and other organic substances can passively cross the Bowman's capsule and into the tubular system. While RBCs, WBCs, and other large molecules that are not filtered will be left in the general circulation. Since the glomerulus is negatively charged, molecules with a positive charge are freely filtered, while those that are negatively charged would repel the charge of the glomerulus and will exit through the efferent arteriole. Ultrafiltration of the glomerular capillaries is influenced by Starling's forces, namely glomerular hydrostatic pressure, glomerular oncotic pressure, Bowman's space hydrostatic pressure, and the Bowman's space oncotic pressure. Compared to other capillaries, glomerular capillaries have much higher filtration rate due to its high glomerular hydrostatic pressure, which should promote water filtration into the Bowman's space. Changes in any of the Starling's forces, specifically the glomerular hydrostatic pressure, which is the primary regulator of the GFR, can alter the filtration of the glomerulus. This is the proximal convoluted tubule. The proximal tubule reabsorbs approximately 67% of filtered water, sodium, chloride, potassium, and other solutes. In addition, the proximal tubule reabsorbs all the glucose and amino acids filtered by the glomerulus. Sodium is reabsorbed by different mechanisms in the first and the second halves of the proximal tubule. In the first half of the proximal tubule, sodium is reabsorbed primarily with bicarbonates. In contrast, in the second half, sodium is reabsorbed mainly with chloride ion. The key element in proximal tubule reabsorption is the sodium-potassium ATPase pump. The reabsorption of every substance, including water, is linked to the operation of the sodium-potassium ATPase pump. Sodium also enters proximal tubule cells by several symporter mechanisms, including sodium glucose, sodium amino acid, sodium organic phosphates, and sodium lactate. The glucose and other organic solutes that enter the cells with sodium leave the cell across the basolateral membrane by passive transport mechanisms. Any sodium that enters the cell across the apical membrane leaves the cell and enters the blood by the sodium-potassium ATPase mechanism. Thus, the sodium reabsorption influences the reabsorption of water and other solutes by the proximal tubule. This is the loop of Henle. It consists of different parts, namely thin descending limb, thin ascending limb, and thick ascending limb. The parts of the loop of Henle have different permeabilities to water and ions, which would function to reabsorb water, sodium, potassium, and chloride ions. As fluids pass through the thin descending limb, water will diffuse into the peritubular space because this section is soluble to water and not solutes. Next is the thin ascending limb. There is no active transport of salt. It is highly permeable to sodium and chloride ions, which cross the membrane via diffusion and enter the tubule, and its permeability to water is very low. Next is the thick ascending limb. The thick ascending limb, on the other hand, is permeable only to ions such as sodium, potassium, chloride, and not water. Due to the different permeabilities of the descending and ascending parts, tubular osmolarity would increase as it passes the descending limb, while it would decrease as it passes the ascending limb. A hyperosmotic gradient is then created in the peritubular space. Water and ions in this space would then be absorbed into the vasa recta, which lie in close proximity with the loop of Henle. This is known as the countercurrent exchanger. The distal convoluted tubule is partly responsible for the regulation of sodium, potassium, calcium, chloride, and pH. It reabsorbs sodium through coupled secretions of hydrogen ions or potassium ions into the tubular fluid, a process which requires the presence of adrenal hormone aldosterone. By acidifying the urine, the distal convoluted tubule plays an important role in acid-base balance. Normally, it is relatively impermeable to water. However, in the presence of antidiuretic hormone, its permeability to water increases, permitting concentration of the urine. It secretes ammonium ions and some drugs, and also forms part of the juxtaglomerular apparatus. The late distal tubule and cortical collecting tubule reabsorb 3% of the filtered water, controls the degree of dilution and concentration of urine. It is also under the control of antidiuretic hormone and relatively impermeable to urea. There are two cell types in these areas: principal cells and intercalated cells. Principal cells reabsorb sodium and water from the lumen and secrete potassium into the lumen via individual sodium and potassium channels. Intercalated cells reabsorb potassium ions and secrete hydrogen ions into the tubular lumen with the use of hydrogen ATPase and plays a key role in acid regulation. The medullary collecting duct reabsorbs less than 10% of filtered water and sodium. This is the final site for urine processing, thus determines final output of water and solutes. Permeability to water is controlled by antidiuretic hormone. Increase in ADH decreases urine volume, hence increasing its concentration. It is permeable to urea and reabsorbs urea through the medullary interstitium, raising its osmolality and its ability to concentrate urine. It also secretes hydrogen against a large concentration gradient. During dehydration, the renin-angiotensin-aldosterone system regulates water balance. When sodium volumes in the tubules is low, juxtaglomerular cells in the kidneys activate the prorenin and secrete renin directly in the circulation. Then it will go to the liver, where the plasma renin converts angiotensinogen into angiotensin 1. Angiotensin 1 is then converted to angiotensin 2 by the enzyme, angiotensin converting enzyme, found in the lungs. Angiotensin 2 is a potent vasoactive peptide which causes blood vessels to constrict, resulting in increased blood pressure. Angiotensin 2 also stimulates the secretion of aldosterones from the renal cortex. Aldosterone causes the tubules of the kidneys to increase the reabsorption of sodium into the blood, allowing water to flow. Also, during dehydration, the antidiuretic hormone is activated. An increase in ADH will promote water reabsorption from the late distal tubules and medullary collecting duct, allowing the body to conserve water.